Good morning, everyone, and welcome to today's Revive webinar by the Global Antibiotic Research and Development Partnership, GARDP. My name is Laura Piddock, and I am the GARDP Director of Scientific Affairs. I have the pleasure of moderating today's webinar. First, a little background for those in the audience who are unfamiliar with Revive. Revive is GARDP's education and outreach programme, and it aims to connect and support the antimicrobial R&D community by facilitating learning, sharing knowledge, and connecting people. These webinars are part of our educational activities. All webinars are recorded in full and can be viewed after the live broadcast on our website revive.gardp.org/webinars. Please visit the Revive website regularly to stay up to date about future webinars, watch recordings of previous webinars, and also to find other information about Revive. Following the presentation, you will have the opportunity to ask questions. You can submit your questions at any time during the webinar via the questions window in your webinar control panel as shown on the slide. We will address the questions after our presentation, and we will endeavour to ask and respond to as many as possible. Before I introduce today's speaker, please note that this presentation was part of a Meet the Expert session enabling academia to fill the discovery gap at the recent ASM Microbe Conference 2019. This was jointly organised by GARDP, CARB-X and the Repair Impact Fund. Today's speaker is Ursula Theuretzbacher. Ursula founded the Center of Anti-Infective Agents Vienna in Austria in 1988. She is an expert in antibacterial drug research and development, strategies and policies based on clinical and public health needs. Her broad therapeutic area of expertise includes early integration of pharmacokinetic, pharmacodynamic, i.e. PK/PD concepts, optimization of dosing and usage approaches, public funding strategies for antibacterial research and development, as well as initiatives to recover the global pipelines for antibiotic drug development. She has been a work package leader or partner in several European Union funded international collaborative projects focused on antibacterial drug research and development and reviving of old antibiotics. She has also served as the president of the International Society for Anti-Infective Pharmacology, founding president of the European Society for Clinical Microbiology and Infectious Diseases, ESCMID, PK/PD of Anti-Infective Study Group, and as an executive committee member of the International Society for Infectious Diseases, ISID. Ursula was a member of the coordinating group of the World Health Organization Project Priority Pathogen List for Antibiotic Research and Development, and is currently a leading scientist for the clinical and preclinical pipeline analysis and development of target product profiles for the WHO. She lectured at the University of Vienna for 10 years and recently became an ESCMID Fellow. She holds a PhD in microbiology from the University of Vienna and from the University of Innsbruck in Austria. Welcome, Ursula. Please start your presentation now.
Thank you, Laura, for this introduction. I will focus today's presentation on the early phases of antibacterial R&D. I will cover the following areas: who is still engaged and who is still active in discovery and preclinical activities. And what are the main approaches globally? Based on these findings, I will present more details about the two big groups that we can summarize as traditional antibiotics or non-traditional therapies. I will also talk about the issues commonly seen in funding applications, learnings from the industry, and finally, recommendations for writing funding applications. So who is filling the discovery gaps? Who is active in discovery and early preclinical development activities? R&D takes mainly place in the so-called SMEs. These are small and medium-sized enterprises. They are mainly based on in-license technology or platforms from academia, or they are founded as university spin-outs. About 85% of all antibacterial discovery and early development projects are based in SMEs. So this is the most important group of discovery and preclinical development activities. Academia is increasingly active in this field, but is focused mainly on basic research and discovery activities. Further, publicly funded R&D institutions are active in this field. Public-private partnerships and non-profit institutions supported by philanthropic organizations, and very few larger companies. They are based mainly in Europe and Asia. These are not the well-known international, globally acting pharmaceutical corporations. What is common to all these institutions? They are usually very small companies or teams. They may have highly specialized expertise, for instance, mainly focused on chemistry. They may overestimate medical need and market potential, but they also may underestimate clinical development challenges. And this high vulnerability is commonly characterized by dependence on one program or one technology. So how are these, what are the R&D challenges by the main discovery approaches? And what are these main discovery approaches? This is, as I said already, the important group of direct acting agents that we call traditional antibiotics. They are usually small molecules. The non-traditional group of therapies can be further divided into several groups. This is the group of potentiators. These are compounds that are potentiating other antibiotics that are usually already existing. Phages and phage-derived peptides usually affect bacteria directly. Antivirulence agents target non-essential gene products. They are blocking a wide range of virulence factors. The antibodies or antibody conjugates, antibody drug conjugates are also researched. Vaccines are an increasingly growing field, and with vaccines, I mean vaccines beyond the well-known vaccines against, for instance, Haemophilus or pneumococci. Microbiome modulating therapies mostly manipulate the gut microbiota. About 20 years ago, we used to call the microbiome normal flora, but now it has the more fancy name microbiome. Repurposed drugs are either non-antibiotics or antibiotics that have been used in a completely different field. Nanoparticles or nanobiotics is also a group that's researched nowadays. Immunomodulators modulate the immune system to support the elimination of pathogens or reduce the negative effects of the immune response. So what are the challenges of all these approaches? I will take some groups and discuss these challenges in more detail. So let's start with the direct acting small molecules. What did I see in lots of funding proposals? The activity of these direct acting small molecules is usually measured by MIC. In the past, commonly these wild type distributions and the MICs in general were very low. That means lower than 0.06 to 0.25 microgram per milliliter. So in general, very low MICs. Nowadays, in many applications, I see much higher numbers of wild type distributions, and of course tested in small numbers. So these higher MICs are currently in the range of 0.25 to higher than 4 microgram per milliliter. So that doesn't mean much if you don't know other factors like protein binding. If a compound has a high binding to plasma proteins and are tested in broth that does not contain proteins, then you don't get the right picture, because protein binding means that only the fraction that's not bound to proteins is active. The consequence of all that, higher MICs and potential protein binding, means that high doses may be required and exceptional PK properties. That's possible, but it increases toxicity problems. Another issue that I could see when reviewing these grant proposals is the spectrum. If the spectrum of a compound is rather narrow, then the question raises, is there a medical need for this narrow spectrum? And is there an indication that would need coverage of these pathogens that are researched? One issue is also mode of action. It's important to know if there's an unspecific detergent-like mechanism present, or if there is a defined mode of action and target. I mentioned already pharmacokinetics. So basic pharmacokinetic studies must be covering the planned use. For instance, systemic use, UTI, or intra or extracellular distribution. If you're planning to target UTI, for instance, because the spectrum enables this indication, then it's important to know that if this drug, if this compound is eliminated by a kidney so that there are high enough concentrations in the urine. So doing the appropriate basic pharmacokinetics that are matching the planned indication is very important. The same is true for intra and extracellular distribution. Some pathogens are localized in cells and some are localized mainly outside of cells. And this must be reflected in the pharmacokinetic studies. Similarly, pharmacokinetics needs to be correlated with pharmacodynamics, in this case, usually measured by MIC. Toxicity signals is a really special topic that has been covered in an extra webinar that you can find on the Revive website. Emergence of mutational resistance is an increasingly important topic. And it's a very delicate topic where it's not possible to really give a cookbook-like recipe what to do. If you would say we compared mutational resistance with rifampicin, and it's great because it's comparable to rifampicin, then I would say you need to do further studies and go into this topic more deeply. So these were some thoughts about direct acting small molecules, and I would like to turn now to the very diverse field of non-traditional approaches. These are antibodies, vaccines, that are usually used for prophylaxis, in prophylaxis in high-risk groups. And that means that you need to know exactly what the high-risk groups are. You have to define them carefully. And it might also mean that these are small numbers of patients that may benefit from this vaccine. So being aware of that is important. If it's used therapeutically, then you would need an active antibiotic or an active compound that you can combine it with. That means it's an adjunctive therapy. The same is true for antivirulence approaches that are usually adjunctive therapies, and you need an active antibiotic to combine it with. The group of phages or phage-derived proteins have been researched since a very long time. So far, they have mainly been used in compassionate use programs and have received a lot of attention recently because we have heard about successful therapies of individual patients. Microbiome modulating therapies are characterized by a complex target. The microbiome is incredibly complex. And they have mainly focused on Clostridium difficile disease. But now a lot of therapies, a lot of approaches are also looking at other pathogens and other disease complexes. Nanoparticles have been used since a long time and also in other fields for delivery of active compounds, but are also now used for direct activity. Immunomodulators is again very similar to a microbiome, a very complex target that we don't understand in all details yet, and they are usually used as adjunctive therapy. So what is in common to most of these therapies? It is adjunctive therapy. That means you have to show that there is a measurable clinical impact and that it's useful for patients. You can show this in superiority design trials. That means that you have to compare with an active arm. So an active antibiotic versus an active antibiotic plus your adjunctive therapy. As traditional antibiotics that are standard of care are usually very active, it's quite difficult to show a difference between these two arms. It's a real challenge to do that. And so far, there have not been much success with superiority trial designs of adjunctive therapies. I would like to go into a little bit more detail now with some of the more common approaches, like the antivirulence approaches. What are the challenges if you are pursuing such a project? It starts with the selection of target. Selection of the most relevant antivirulence target according to their contribution to disease is a challenge. Virulence factors are not expressed uniformly in all strains. They may differ geographically, and the genes may not be present in all strains. So you have also a genetic variation. The consequence of this challenge is that a big number of strains have to be tested to know how these virulence factors are distributed. The virulence factors may be effective only in specific disease states, for instance, in chronic or dormant bacteria, or they may be only effective in specific time points in the infectious process or at a specific infection site or in specific patient groups, for instance, immunocompetent patients. Another challenge is that diagnostics are needed. And these diagnostics are needed beyond species identification to guide the use of antivirulence drugs or therapies. And this diagnostic availability is not existing today, so it has to be developed. One of the really biggest challenges is dose finding. In traditional small molecule antibiotics, we have predictive models, PK/PD models, that are quite effective and predict clinical outcome. And this is not existing in antivirulence approaches because usually we don't have an MIC that can go into these traditional PK/PD models. So to explain this further, we don't have these validated models to predict clinical outcome. And the current preclinical studies may not be meaningful to predict clinical outcome. So that means even if we have very promising preclinical results, it does not mean that it can be translated into a clinical effect. In general, it may also not be known if the selected approach has sufficient clinically relevant impact on the disease in humans. We just don't know it yet. And as I have said already, superiority studies are essential to show that the adjunctive therapy provides benefit to patients. This is not only necessary for regulatory purposes, but also just for usage to guide clinicians. Superiority studies and also preventive studies are very difficult to do. So the next field that I would like to explain is microbiome modulating therapies. Manipulating and engineering the human microbiome is seen as attractive option to influence a broad range of diseases, including bacterial infections, and is very popular among investors. And we have seen some spillover of this excitement from investors and also companies. There are some trends seen recently in this field. You may all know fecal microbiome transplants that are transferring entire microbiomes from healthy people to patients. The trend now is reducing the complexity of microbiomes. Based on new techniques, genetically engineered probiotica to express different antibacterial proteins or specific metabolites are pursued. It's very scientifically interesting, and it's a real advance to use these new techniques that are based on systems biology and synthetic biology. Also using phages as vehicles for antibacterial compounds are commonly used. What are the challenges of microbiome modulating therapies? The reduction of this complex ecological system to selected components of the microbiome is maybe a challenge, and we just don't know yet if that also reduces the clinical outcome. We don't have predictive models again, and if this approach is intended for therapy, then it still is an adjunctive therapy. And we have all these translational challenges that I mentioned before for the clinical development. So in general now, I've listed here some translational challenges for all these preclinical programs. Quantifying the medical need realistically is very important. Doing all this effort of a preclinical program and then discover that your great approach and your program is not really needed is not a good experience. Developing and following a target product profile is very important. So what is a target product profile? It summarizes the desired R&D outcome, and it serves as internal guidance that should prevent insufficient quality of the program. These TPPs should not be adapted to the program, but the program should always follow the TPP. So developing TPPs is very important. Toxicity signals are very important. It's very difficult to give here general recommendations. So just follow general guidance. And if a signal is noticeable, then explore it. Just don't ignore it. PK and PK/PD and potential dosing is more important than you may think. Sometimes I see very small therapeutic windows. So the required dosing, the required concentrations are so high that the toxicity is not far away from the therapeutic concentrations. And this is not a good situation. If MIC does not apply because you have an approach that's not based on MICs, then development of predictive animal models is most important. For some approaches, like some biological approaches, CMC, that's chemistry, manufacturing, quality control, may be a very specific challenge. For the clinical development, enrolling enough patients for the chosen indication. Are there enough patients available for clinical trials for this indication? Are there enough patients available for testing resistant pathogens? Is the resistance rate high enough to find patients for enrolling in clinical studies? And for adjunctive therapies, consider superiority design of clinical studies with all their challenges of clinical studies. What we could learn from industry, broaden expertise to all relevant fields is very important, especially in small companies where you cannot have all expertise in the company. So the choice of advisors with a broad range of expertise is very important. The next advice, I know it depends a lot on available resources that may not be available in small companies, but try not to take shortcuts that may jeopardize the program. So this may be really a high risk if shortcuts are taken. Structured, informed go/no-go decisions can really help very early on to take good decisions. And consider partnerships and collaborations. So finally, I would like to give some recommendations and how you could prevent annoying reviewers. Reviewers usually have to read a lot of text and hundreds of pages, and it's important to help them understanding your projects. So if you are writing proposals, then there are some advice that I could give you. So try to have a congruent concept and structure. That means the reviewer should always know what you are writing about. If you have to go through hundreds of pages and still don't know if the information is about historical aspects of your project or the current situation, then maybe the structure could be improved. Address challenges, gaps, and lack of experience. Describe killer experiments and go, no-go decision points. Reviewers get usually very suspicious if the project is described only in glowing terms and highest praises. So be honest, that's more important. And if there are challenges, and there are always a lot of challenges in these early projects, then address them and describe them. That's one of the most important recommendations I can give you. Some important issues that you should address are protein binding, wide MIC distributions, potential cross resistance, emergence of mutational resistance, in vitro and in vivo models, if they are not established yet, and clinical development path. Avoid copy-paste style and extensive non-essential information. If you have to go through a lot of pages with information that's really not essential for this project, then it may annoy reviewers. Label figures correctly so that the reviewer really knows what this figure means. As I've said already, be honest, that's much more important than being over optimistic. If the funding round is organized in 2 steps, then give enough information in the first non-confidential round. If this information is not good enough to know what the project is about, then you will not get into the second round. Provide a clear summary that really helps. So with this, I hope I could give you some insights from my experience as reviewer of a high number of R&D funding applications and my assessment of global antibacterial R&D pipelines. With this, I'm handing over to Laura. Thank you.
Thank you, Ursula, for an excellent presentation. We will now start the question and answer session. As a quick reminder to the audience, please send your questions via the questions window on your webinar control panel, and we will ask and respond to as many questions as possible. So the first question is, what are the main criteria for attracting the interest of funders?
Yes, that's a good question. So one of the main criteria is, does the project contribute to solving a significant public health and medical problem? Usually that's addressing a resistance problem. Is this project novel, or is it me too? Is it just a small optimization of something existing? This is usually linked to resistance. Is it really meaningful? Does it do something for a patient? Very important is also, is the team competent? Do they really address the relevant questions? Do they know the hurdles and challenges? Do they have a plan, or did they at least think about how to develop their product and how it can be used clinically? And as I have said in my last slide, is the proposal written in a way that makes a realistic assessment possible? Yeah, and lastly, look carefully at the scope of the funding call. It does not make sense to invest a lot of time with writing of a proposal if there is no fit. So all this really helps to attract the interest of funders, but of course it depends on the funder, and there are very different criteria that would fit to different funders.
Thank you. We have just had a question that can be addressed by listening to a previous webinar. So I will read the question to you, but not expect you to answer it, Ursula. So the question is, what is the criteria for selecting an animal model? So the person who's asked this, please would you look at our previous Revive webinar on in vivo models? Thank you. The next question for you, Ursula, is if you were a young investigator starting an independent laboratory working on antimicrobial resistance, what indication and approach would you recommend they focus upon?
There is a good guidance, I think, and that's the WHO priority pathogen list for R&D. That guides the main interest nowadays, and this is really gram-negative pathogens with high resistance rates. So that's the highest medical need. But it does not mean that other pathogens are not important. But if you are targeting other pathogens that are not really the main priority, then the project has to be really excellent science, new class, new target. So it's really not so much the indication. It's can you target different groups of bacteria? Targeting gram-negative bacteria has its very specific challenges, and we had also webinars about this topic. So you have all this penetration and efflux challenges. But this is a very attractive field, and there are several indications that can be targeted with gram-negative bacteria, from UTI to intra-abdominal infections to hospital-acquired pneumonia. They are all very well developed regulatory path and clinical study design. So this would be good indications, but of course it all depends on your previous research experience.
Thank you. The next question is about antivirulence compounds. What current antivirulence compounds are in clinical trials or in use in clinics?
We have some approaches even in clinical use, and for instance, these are antibodies that are usually targeting toxins. So an example would be Clostridium difficile toxins, but also Clostridium botulinum toxins. So toxins that are usually causing the symptoms of this disease are targets that have been pursued by antibodies. And there are some, very few, but some are in even in use, and more are in clinical development. Also microbiome modifying, but these are not antivirulence approaches, are also in clinical development. Otherwise, antivirulence approaches are usually in early stages of development.
Thank you. It's important to mention to the audience that indeed, Ursula, you and I have just had a review published in Cell, Host and Microbe on non-traditional approaches, and there's further detail in that on the current approaches under development. So moving on to the next question. You mentioned at the beginning of your presentation that companies involved in drug discovery are not always well known. For academic researchers who wish to collaborate or partner with some of these, how would they find out about these companies?
This is a really difficult question. Because there are so many non-confidential issues and confidentiality issues around these programs, that it really takes a lot of effort to find out which companies are active in this field. So one option would be looking at the programs of partnering meetings. Usually you find there are some companies that you didn't know before, or you look at poster sessions of conferences, and then of course the literature. This is an important issues, and some organizations try to help with these collaborations and partnering questions. I can point you to a current project of WHO that tries to identify these companies that are willing also to give their information to the public. And I would guess that at the end of the year, you will find this preclinical pipeline analysis, but that I am contributing to on the website of WHO, that will list companies with their projects, at least as far as they have public information available.
Thank you. Should a program be continued if activity beyond that for gram-positive bacteria cannot be achieved?
Yeah, so you may all know that gram-negative bacteria present a very specific challenge, a challenge of a complex outer membrane, and penetration, efflux are scientific hurdles. That means that many programs, especially in the past, focused on gram-positive bacteria. And the question is, it still be pursued despite the need is more focused on gram-negative bacteria. I would say that a gram-positive program is still viable, but it should be a real novel, promising approach. That means a new class, new target, so no modification of an existing antibiotic. With an exciting science, a competent developing team, and what would also be better is if it's not a pathogen-specific approach. That means Staphylococcus aureus only, for instance. It would be better to have a broader gram-positive spectrum. And in these cases, I would say, yes, gram-positive projects are still worth pursuing and will be needed, especially in the future.
Thank you. Continuing with that theme, is there a need for pathogen-specific drugs? And if yes, any specific pathogens which are most important to cover at the moment?
Yeah, I would say that there's no specific need for a pathogen-specific approach, but there are situations where pathogen-specific drugs may be useful and also acceptable. In any case of a pathogen-specific drug, there's the need of a diagnostic confirmation that can confirm the potential usefulness in a specific situation. So pathogen-specific approaches could have been used and are still used for Pseudomonas or, for instance, Acinetobacter, because these are very critical pathogens with partly high resistance rates. Other options are infections that are typically caused by one specific pathogen. An example is Neisseria gonorrhea or Salmonella typhi or Mycobacterium tuberculosis. So in these cases, of course, pathogen-specific approaches make a lot of sense.
Thank you. What should be done if traditional predictive MIC-based models are not applicable, such as for agents under development as antivirulence therapies?
There are mainly, or I see mainly two options. One is just go into clinical trials without knowing if your therapy works. Try superiority trial design, but this is a very high risk. You just don't know what happens, and a high risk of failure. That's more the approach that we used decades ago for the first antibiotics. Just go into clinical trials and try and see. So the second option means a lot of work and investing in developing new models that may include validated biomarkers or surrogate markers. They all have to be really clinically validated. It means a lot of work that still needs to be done before you could or should go into clinical trials.
Thank you. This is following the theme on antivirulence compounds. So there has been much interest in academic laboratories on exploiting virulence as a target for new antimicrobial approaches. The question is this an academic fashion, or are drug developers and funders of drug development really interested in funding and supporting this work?
This is a good question. Um, I think that it's an approach that it's very common in academic institutions, and I fully understand the reason. It's just very scientifically interesting. And you have so much publications about antivirulence and virulence factors. It's really scientifically interesting. You don't see that in bigger companies. You have not seen that in big pharma before. Um, and funders may also see it as a more basic research funding. Um, um, but in general, I think that the hurdles are very high, and these are very high risk approaches. Funders tend to have very specific calls that are very specifically focused on either direct acting drugs or other approaches. But we don't really know. It's very hard to predict if any of these approaches will be clinically valuable in the future and have clinical impact on patients. It's much too early now to say if these approaches, and I'm talking now really in general, there may be some exceptions that we have seen for antibodies against toxins.
Thank you. This next question follows a similar theme. How much risk are publicly funded or philanthropic funders willing to take in supporting antimicrobial R&D approaches?
Yeah, funders are really now the main source for supporting small companies and also academic institutions. There is not much private investor money now in this field. So it's very important what funders are doing. And it's a big, of course, big difference between a funder and an investor. So this question really depends on the funding organization and also on the program. I think if the proposal, so the program, is reasonable and addresses a major challenge, a major clinical challenge, with plans how to cope with them and cope with the challenges of the program, a funder may be more willing to take a risk, because that's what funders should do. They should fund programs that would otherwise not find investors because of a risk. So to summarize, addressing an important public health issue and excellent science may attract interest of funders.
Thank you. The last question is another more philosophical question for you. So your presentation has essentially covered what academia and small companies can do, left by the gap of large pharma. You've also suggested that people should work in partnership or in collaboration. How could you make the link between these and fill the gap between academia and small companies and large companies who will ultimately do the final development on the drugs?
Yeah, this is a really big question with many different aspects. There are now increasingly institutions that are mainly focusing on collaborations. It's very difficult to do everything by yourself, and even the clinical development by a small company is a real challenge that has been achieved by some companies in the past, but it's still a challenge, and collaborations are very important. There are a lot of projects funded, for instance, by the European Commission. They are bringing together academics and companies. And there are also institutions like GARDP that is based on collaborating with all kinds of different institutions. And this concept has also been taken by bigger companies that are mainly collaborating on many different projects with specialized other players. So this concept is, I think, very important, and maybe even the future of antibacterial R&D. It's since Big Pharma has left this field, it's a challenge to fill all these fields that are necessary to bring an antibacterial therapy from discovery to the patients. And we'll see how we'll do it in the future.
Thank you. So we have now come to the end of our question and answer session. Ursula, once again, I thank you for your presentation and for providing your valuable insights during this presentation and Q&A session. I now will hand back to Astrid Pensmuir at GARDP. Thank you.
All right, thanks Laura, and thanks Ursula for your presentation. I just want to still take the last minute to point out our next two of our future webinars. On the 20th of August, we have Lena Friberg and Elisabeth Nielsen from the Uppsala University in Sweden, who will present in a webinar on computational modeling for population PK and PK/PD. On the 3rd of October, we will have Olga Geniu from the Fundación Medina in Spain, who will speak on a webinar about traditional screening and novel discovery approaches for natural product antibiotics. You can already register for these two webinars on Revive. And also watch out for another webinar on in vivo models, which will be coming in September, and I hope to be able to share more details about this soon. With this, I would like to thank everybody in the audience for joining today and for contributing to the discussion. I really hope this webinar was interesting for you and useful, and I hope you will join us again for our future webinars. Thank you and goodbye, everybody.